Okay, well, welcome to your lesson on the parts of the animal and plant cells. I'm going to run through them as quickly as we possibly can. First of all, a lot of this stuff should be familiar from grade 10, hopefully. You should know the nucleus. If I asked you what the mitochondria does, your elementary school teacher told you it does what? It is the powerhouse of the cell. Okay, so it's something to do with energy production. We will focus in on exactly what that means when we get to the next unit, when we talk about metabolic processes. So we'll get to that, and we'll go specifically and say, how exactly does the mitochondria help the cell to burn, in quotation marks, burn sugar? So we'll go through that, and how do we get energy out of that? So ones you should already know then would be the nucleus. In grade 11, you learned about DNA, chromatin stuff. Do you remember the difference between DNA and chromatin, though? Like, why do we ever call it chromatin? Obviously, that's what the spaghetti is. And also, I should say, caveat, this is an artist's conception. It's a cartoon, so it's not going to be perfectly done. In fact, the microtubules, which is this guy here, and the microfilaments are done so poorly in this particular thing. So I'm going to show you a much better animation of what it really looks like on the inside of the cell. Keep in mind, that's the spaghetti also is supposed to represent the chromatin, but it's a cartoon, so be generous with what it looks like. Let me pull up some pictures. So the DNA molecule itself is the strand, and it is the order of bases, A, C, T, and G. So when you look back at that helix structure that we saw before, the ladder, this is the ladder with the bases on the inside. So it would have looked like something like this. That's DNA. Deoxyribonucleic acid. It's the nucleic acid, so it's the polymer version of all these little nucleotides. And of course, the nucleotides would have a phosphate, a sugar, and a base as one piece. And then you have another nucleotide, another nucleotide, another nucleotide. When we get to this structural level, and we're going bigger and bigger and bigger, just like with the proteins, we started with a chain of amino acids. And then based on what was dangling off of them, they would either coil up or they would do these pleated sheets, and then there'd be side chain interactions, and now all of a sudden you've got yourself secondary, tertiary, and then finally quaternary structure when you put everything together and you put all the ions in the right spot. Now you've got a functioning protein, right? Well, DNA is a lot more complex than just saying A, T, C, and G as well. So we start off with the DNA molecule itself, about 2 nanometers, and I want you to picture how big a millimeter would be on a ruler. Then I want you to cut the millimeter into 1 million pieces. Now you've got a nanometer. Just take 2 of those pieces. That's how wide DNA is. It's extremely small. DNA by itself, as 2 nanometers wide, and as being such an important code, is actually quite fragile. You don't want it to snap in any way or anything like that. So every organism, they're all DNA based. Every organism will have some way of protecting and making sure that their DNA is packaged safely so that it can be used safely without damaging it. You want to limit the damage. So the first thing that it will do is it will take that DNA strand and wrap it around these proteins called histones. There's actually 8 of them in this lump here. And it'll wrap it very carefully around that. And now it's about 11 nanometers wide. But then when you coil those guys together, now you have chromatin. So it's significantly wider and larger than the DNA molecule itself. It's a well-protected strand of DNA. That's the spaghetti that we see in our other picture. Then after that, of course, then you do some more folding, and then you fold again, and you fold again, and you fold again. The only time you would see this huge big structure here, this actual chromosome structure, is during replication, when there's a reason to coil it up because I have to package it and move it. Otherwise, it's always going to be in this looser chromatin, which again is what our cartoon was trying to show as the spaghetti that was inside the nucleus. So that's what we're looking at when we're looking at this spaghetti that's in here. That's the chromatin itself. So it's well-packaged DNA, but it's a looser strand than what you would get if it was a chromosome. Okay? It's so thin, that 30 nanometers, that we cannot see it on the micro scale. It's on the nano scale. 1 nano is a billionth of a meter, whereas micro is only a millionth of a meter. So it's an order of 1,000 difference between what we can see here in the lab. In the middle, so the nucleus would be, obviously it's cut open in this picture, the nucleus would be the protective package for that really important DNA that we don't want damaged. Partially because out here floating in the cytoplasm, there are enzymes whose job it is to recycle nucleic acids. And I certainly don't want the enzymes attacking my own DNA. That would be a problem. So I'm going to carefully package it, hide it away, and it's going to be safe inside the nucleus. These little holes here are called nuclear pores. They're not technically their own organelle. They are a part of the nucleus. In the middle of the nucleus itself, there's this darker dot that you can see when you're looking at through the microscope itself, looking at an actual cell. And that darker dot is called the nucleolus. Not the nucleus, but the nucleolus. And the nucleolus is important for ribosome production. So whereas the DNA has the codes for making literally every other protein in the body, in the cell specifically, the nucleolus is in charge of making sure that the ribosomal RNA and all that kind of stuff is ready to go. So it's involved with ribosomes. As we move outside of the nuclear membrane and beyond the nuclear pores, the pores are going to be there for the sake of inside the nucleus, I have my DNA. I never want my DNA to leave because I don't want it to come out and be damaged. So instead, I take a snapshot of the one piece of the code that I want, and that snapshot is called RNA. mRNA specifically, it's called messenger RNA, and it will be created inside the nucleus, just a little snapshot, a replication of that particular section of DNA. It will leave out the nuclear pore, and it will either go to one of these free-floating ribosomes to be read and created into a protein, or it'll go to the endoplasmic reticulum, again, where the ribosomes are attached. If you have endoplasmic reticulum with ribosomes attached, you call it rough endoplasmic reticulum. And if there is no ribosomes attached, you call it smooth endoplasmic reticulum, literally because of what it looks like. The term endoplasmic, though, let me just go back to that word. Endo means inside. Inside where? Well, we're inside the plasma, cytoplasm, right? Plasma. And to reticulate, this is actually a term that you can use in music. When music comes back to the theme, the original theme, so you have the music, la la la, so let's say verse, chorus, and then you're back to a verse again. That's called reticulation. You have folded back on itself and come back to an original theme. So the idea of to reticulate or to have a reticulum means that it's folded. So that's it. You're looking at these membranes, and the membranes are folding back in on themselves. That's why. So it's actually just the fancy name is just describing what it looked like when they looked under the microscope and they saw it for the first time. It's like, oh, look at that reticulating thing. It's in the plasm. So they're like, oh, we can't say it's in the plasm. That sounds dumb. So someone else said, oh, how about endoplasmic reticulum? And then somebody's like, that's brilliant. Let's trick everybody and pretend like it's hard. That's what they did. Okay. So then you have the endoplasmic reticulum, again, studded with ribosomes. Means that they are involved in processing, creating proteins, okay? Not protons, but proteins. Not studded means they're probably not so involved with proteins. In fact, we think that the smooth ER is more involved with processing fats instead. Now, smooth ER can also be packaging proteins as well because they're all connected. They're just a huge big channel, an inside channel where we can have these proteins being created and then moving through the channel to be packaged. When I package them, I'm actually packaging them in a piece of the membrane, and it'll bulge out of there and form something that looks like where's one? Not these guys with the little circles, but imagine another one that didn't have the little circles around it. It would form a little, kind of like a little vesicle here, and then the little vesicle would float up to the Golgi body, or the Golgi apparatus, your choice on what you call it, for further processing. We've already said that with the proteins, you have all those different levels of organization, right? So when we go back up to that note, when you go here, you say, okay, well, you have to put them in order, get the right order so that you know whether it's going to coil up or pleat. The order, we said, was done by the ribosomes. And then the place where the folding happens is going to happen either within the cytoplasm, because that's where we have some free-floating ribosomes, or it'll happen in the endoplasmic reticulum. Okay. Then it gets packaged. The different subunits and whatnot can then be sent, kind of like FedEx within the cell, can be sent directly to the Golgi body. And the Golgi body does the final job of making sure everything is put together properly. And now the protein is active. It's able to be used. So then either the Golgi body will repackage it again and send it out to the cell membrane to release it from the cell, or it'll just be carried to somewhere within the cell where it needs to be used. It depends what the product is. Is this the type of cell that's secreting stuff so that the rest of the body can benefit from it? Well, then it's going to deliver it to the cell membrane and release it. So these little bumps in the cell membrane here that you're seeing, they're trying to show that we have vesicles like vehicles. Vesicles are being sent up to the cell membrane. They're fusing and becoming part of it and being delivered to the outside. So that's the connection between the DNA, where you have the code itself. The RNA will come out the nuclear pore. It'll be read by a ribosome. The amino acids will be put into the correct order. Within the endoplasmic reticulum or the cytoplasm, which is just the jelly, you will have that protein folded, and then it can be packaged into a vesicle, sent to the Golgi body in order to be modified for whatever it needs to be modified with. You got a hemoglobin, you want to stick the subunits together and add some iron to it? Great. Golgi body can take care of that. And then the Golgi body can either deliver it via a vesicle to the outside or to the inside. The Golgi body is also going to be responsible for helping to create these right here. It's in blue down here, and they're all over the place. It's the ones that have a double membrane, but they seem to have these other little smaller things attached to them. These are your lysosomes. A lysosome's job is to lyse, L-Y-S-E. A zome just means body, a thing. A thingy is really what zome means. Okay. When they saw chromosomes for the first time, chromo means color, zome means body or thingy. So they're like, hey, look at all these little colored thingies. And then a guy in the office next door was like, you cannot call them colored thingies. What are you going to call them instead? They're like, oh, okay, fine, chromosomes. And again, making it fancier than what it actually is. But they're literally just describing what they see. So when you see lysosomes, they're describing the fact that the lysosomes are basically like the garbage collector and destroyer of the cell. They have enzymes inside called hydrolytic enzymes. Hydrolytic enzymes perform hydrolysis. What's hydrolysis? Digestion, right? Adding water to something to break it apart like you would in digestion. So these sacs that are created, these lysosomes, contain hydrolytic enzymes whose job it is to go around and kind of mop up the dirt. They don't just destroy waste, they also will go around and start to digest things for the cell itself. So the little tiny things that are attached to it are just little tiny vesicles that may have come into the cell or may have come from somewhere else within the cell. And they contain food, waste, all sorts of different things, whatever needs to be digested. And it'll be, the lysosome will attach to it, suck it in, and then go for it. Now you're going to have in your homework 2 terms. You're going to have primary and secondary lysosomes. A primary lysosome is just one that's not working right now. It's turned off. A secondary lysosome is after it is attached to something that it's going to eat or that's going to digest. Now it's called a secondary lysosome because it's active. So don't get tripped up on those things. Just know lysosome in general has a bunch of these digestive enzymes inside, hydrolytic enzymes. Okay, so these are the enzymes themselves would have gone through the same process as any other protein because they are proteins. They would have had a code somewhere in the DNA. mRNA would have come out, ribosomes would have read it, put it together, would have been packaged, sent to the Golgi body. Golgi body would have made sure that all the right enzymes were in the right place at the right time, and then it would have released this as a vesicle, and the lysosome would have gone off and done its job. So it's all connected that way. Again, this is artist's conception, so it's not very well done in this particular diagram, but you have these tubes, which are called microtubules, and then you have these little guys here that are called microfilaments. And I'm going to show you a video that really does a much better job of trying to show what they're for and kind of why you care about those. They're going to be structural, though. They're going to be holding the cell together. They're going to be a pathway for these amazing little kinesin motors to drag things along and move things around. They're going to be all sorts of stuff. They're structural, and they are functional highways for the cell, you'll see shortly. So this picture does no justice to them. But before we move away from this picture, let's talk about some of these other little things that are in here. So you've got your mitochondria. Notice the shape of it, the way it's folded as well. You're going to find that over and over and over again in living things, they take these massive structures, and by folding them, they're able to take them and put them into a much smaller space. It would be like the sheet on your bed takes up a lot of surface area, but when you fold it, it's easy to throw in the cupboard. It's still the same surface area. Now imagine that along that surface area of the sheet on your bed, you've got all these machines that are doing their job. So now you've taken all these machines and you've packaged them and folded them into a small area, and they don't, it's a 1x1 square rather than a, queen size beds are what, 6 foot? So 6 foot by 6 foot square packaged into a much smaller space. So folding is really important for increasing surface area, but reducing the amount of volume it takes up. Brilliant. You're going to see this all over the place. It's why you have folds in your brain. It's why there's folds in your intestine. It's why there's folds everywhere. Folds in your stomach. Folds down here at the cellular level as well for the purpose of nicely packaging things. Part of the structures that you wouldn't necessarily see all the time or care about all the time would be something like this little double log structure right here. Together, the two logs are called a centrosome. Individually, these two logs are called centrioles. Centriole is probably how it was described to you back in grade 10 when you studied mitosis. So these guys are involved. They will double up and then split and move to opposite ends of the cell when the cell is going through division. And out of them are going to grow spindle fibers, which look a lot like these filaments here. And the spindle fibers will, once everything's already organized, whatever, you would have already had the chromosomes condensed back into, so chromatin would condense to chromosomes, so you have these huge, big, fat structures. And then out of this would grow these little spindle fibers. They would attach to either side of that X structure and pull them apart. And they would shrink as they pull them and pull them to opposite cells. And then you'd have that ratcheting in of the cell membrane. And boom, I've got two cells now. With all of these parts in both, because that would have doubled up as well. And now you've got two cells that are functional, and each cell has the complete code to function as they should. So centrioles are involved in cell replication. I want to switch now and show you what it looks like with better animation. So what we're looking at in this particular picture is the inside of a blood vessel. So you're in it like a capillary or something. Okay? These big bluish white guys rolling along are the white blood cells, and you can see red blood cells floating past really, really quickly. What you're going to see is these big cells, which are the epithelial cells that line the inside of every tube. Because there are cells there. There are actual gaps in between cells as well. And so the liquid can leak out. Plus, these guys who are actually able to seek and search and destroy, they're able to dig their way in, especially if it's swollen. So if you've got, there's a chemical called histamine, and histamine's job is to cause inflammation. And inflammation is basically just swelling of these cells. And if these roundish cells get more swollen, it opens up the gap in between and allows the white blood cell more space to squish its way in and fight the bacteria that might be on the other side of this lining. So that's inflammation caused by histamine. That's why if you have allergies, you take a drug called antihistamine, because you don't want that swelling feeling in your face when it feels like your face is blowing up. What these guys will do is they'll have on them these little kind of like high five markers as they're walking along. Every cell on the outside of them has all of these amazing little proteins. They're called glycoproteins and glycolipids. We'll look at that in our next lesson a little more detailed. But these little kind of receptors on the outside of their cells. And those are going to identify and say, I'm this type of a cell. So I'm a liver cell, or I'm an epithelial blood vessel cell. And I belong to so-and-so. And if either of those markers are wrong, then the white blood cell can say, you don't belong here. You're foreign, and they have this little connection that happens, and then the white blood cell can actually literally eat that foreign cell. So that's what it's doing with bacteria all day long. It's meeting up with a bacteria and saying, hey, you shouldn't be here, and it'll attack it and eat it. Same thing with your dead and dying cells. Sometimes cells will throw out a little SOS to the outside of their cell and saying, I'm at the end of my life, can someone please come and clean me up? And then the white blood cell will come and do that as well. In this case here, what we're going to see is the glycoproteins and the glycolipids interacting, rolling along, checking to make sure everything's fine. So here we have them zoomed in, and you can see here's the little interaction between them. Now, sometimes as well, you could have a hormone that could be coming along and telling a cell to do something. Either way, you've got something that's going to happen now on the inside of the cell. This is a type of protein that is actually held together with another protein in what we call a cholesterol raft. We mentioned very briefly cholesterol. Actually, I don't even know if I even mentioned it. It's on your page for lipids. Cholesterol is actually produced by your body. You make all the cholesterol that you need, so you don't really need it in your diet. It's not essential for your diet. But you're using cholesterol for all sorts of things. You're using cholesterol to make sure that your cell membrane doesn't melt and that it doesn't freeze. And you're also using it for these types of purposes as well, where you're kind of building these structures to keep these proteins together. It's sort of like if you had a huge factory warehouse and you had one part of the machine maybe had to do a stamp on a piece of metal, and then you had to take that piece of metal and add it to another, I don't know, like the car door or something. If the car door and the car itself are in two completely different parts of the factory, you're not going to be very efficient. But if I can put those guys working side by side, Then you stamp the car door, you throw it on the car. You stamp the car door, you throw it on the car. And that's what the cholesterol rafts will do. They'll put the two machines in the same place at the same time, so it's very efficient. So this guy makes a product that guy then works on. Okay, so you see all sorts of interactions. Here's a protein built into the membrane. Here's the actual microfilaments that are attached to some of the pores on the surface. Now we're on the inside of the cell. So you can see it's way better to look at something like this. It's very organized structure compared to what we saw. Here's another set of microfilaments as we're running through, holding things together, giving us structure. This is how a microfilament is put together. It's these little subunits of proteins that just get put together, and we can also get rid of them as well. So we can build the fibers and we can remove the microfibers. Here's an enzyme coming to cut a fiber. Okay, wraps around, so it had to be the right shape. This is a microtubule. Notice how it's also built of these little subunits. You can see the subunits, now it's being eaten and dissolved away. This is one of my cool favorites of all of the things here. This is called a kinesin motor. It's also a protein, but its job is to use the microtubule highway and use energy to drag the vesicles around. Let me pause for a second here. In this picture, what we can see is a whole bunch of these microfilaments and microtubules kind of without. Here, do you recognize the structure? Two logs on top of each other? That's the centrosome. Each one is a centriole, and out of that come the filaments, the microtubules and whatnot that are going to form our spindle fibers. Right? Over here, this weird slug-like thing on the right-hand side, that's a mitochondria. And in the background, you can see that there is endoplasmic reticulum being drawn here. And this is a vesicle. Let's keep going. Okay, these are the little pores. Let me pause again. This is the nucleus itself with the nuclear pores. And out of the nuclear pores, you're seeing little pieces of RNA popping out, specifically mRNA or messenger RNA. This is the snapshot or copy of the DNA that was on the inside of the nucleus. And it's going to go out, and there's going to be these ribosomes out there that are going to attach to it. You'll see them coming from the right-hand side now. Two subunits together. They attach. They read it. This is the ribosome. And what you see now is here is the RNA genetic code wrapped with the ribosome. And inside the ribosome, they're grabbing amino acids and putting them together in the right order according to whatever the code said to do. So do you have to grab a glutamine or a lysine or whatever it is? They're grabbing them all in the correct order and building out that polypeptide on the other side. And then the polypeptide, depending on what's dangling, will form helix coils or beta-pleated sheets, and it'll all be put together by the Golgi body in the end. Okay. Just more proteins doing their jobs. There's a mitochondria in the background again. Okay, here, in this case, the ribosome actually attached itself to the endoplasmic reticulum, so the protein is being made inside. And here we have the animation of parts of the endoplasmic reticulum. Oh, there's my little friend again. Parts of the endoplasmic reticulum are now forming vesicles, and these vesicles are going up to the Golgi body now, becoming a part of it, delivering their payload, and the Golgi body is then making new vesicles out the other end that, again, can either be let out to the outside of the cell, like what you see here, or become part of the cell membrane itself, et cetera, et cetera, et cetera. So that gives you, I hope, a better picture, at least of what microtubules and microfilaments are supposed to look like on a better scale. So now these are the proteins now built into the cell membrane that are now available to come and talk to this cell here. It's a complex process, right? Like it's not, we kind of made cells look like, oh yeah, there's stuff inside in grade 10, but there's a heck of a lot going on. It's actually even way more complicated than obviously what we'll do in grade 12. Okay, so things are a lot more complicated than what it appears at the surface. One of your jobs will be not just to be able to label and not just to be able to say this is its individual function, like ribosomes creating peptide bonds for amino acids in order to create polypeptides. That's not the only thing you want to do. What you also want to do is make sure that you know how they work together. So I've already said it a few times. I'll say it one more time. The nucleus holds the DNA code that can then send information out to a ribosome. The ribosome reads the RNA, and it's going to create a polypeptide from that. It can either do it free-floating within the cytoplasm, or it can do it while attached to the endoplasmic reticulum. If that's the case, the endoplasmic reticulum can then create a vesicle, okay? And the vesicle can go to the Golgi body. The Golgi body can process the protein add in whatever it needs to do, put the subunits together, make it a functional protein by modifying it, and then create its own little vesicle and either deliver the payload to the outside of the cell or to wherever in the cell it needs to be released to. So you can talk about all those different organelles all in order, and what I want you to do is be able to say, this is the job that they're doing, whether it's protein synthesis, making proteins, or whether it's delivering and secreting things to the outside of the cell. You can talk about a few of these guys in a row saying this is what they do together. You're going to find that it's actually easier to mention 5 or 6 of them than it is to only mention 3. So even though the requirement might say mention 3 or 4 in what they do, you're going to need to, in order to have a complete conversation, you're probably going to have to do 5 or 6 of them. But it's what I've just said here. It's that simple. How do they work together? So make sure that you're not just able to label, not just able to match functions, but be able to talk about what they are doing together to get a job done. You're also going to need to know what is unique for plants and what is unique for animal cells in terms of their organelles. And for animal cells, they have a centrosome. So this is involved in mitosis, right? But plants don't have this. They have another way to get their spindle fibers going. They don't have visible centrioles. Plants do not have a lysosome, but animal cells do. And then vice versa, the plant cell has the ability to produce its own energy. So the plant cell has a chloroplast. We'll learn about that in our next unit as well, exactly how it works. How does the chlorophyll actually help us to make sugar? We'll tell you all about that. It also has a huge, big central vacuole, also called a water vacuole in some cases, and the fancy word is a tonoplast. Make sure you're able to recognize that that's the same thing. And then, of course, it also has a cell wall. Again, back in grade 9 and grade 10, you would have heard that animal cells don't have a cell wall, plant cells have a cell wall, and they kind of made it sound like the plant cell's the only one that has a cell wall, when in reality, the only one that doesn't have a cell wall out of fungus, bacteria, plants, etc., any other kingdom, animal cells are the only ones that don't have this secondary cell wall. The unique thing that plants have compared to animals, but not plants compared to everything else in nature, if that makes sense. We've already talked about the endoplasmic reticulum. We've talked about the mitochondria, powerhouse of the cell. We're going to have cellular respiration happening there. In this particular diagram, where is it? This here is considered the rough endoplasmic reticulum, and then this down here is considered the smooth endoplasmic reticulum. Again, this is a cartoon. It's artist's conception. It's wherever they could fit it on their diagram, right? And then on this diagram as well, this here in what I've highlighted in red is the chloroplast. When you're choosing to label whatever structure you're going to label, please do not ever choose this part of the cartoon to label. Even though technically it says it's F, technically it says it's a chloroplast, it's such a junky drawing of it. Pick the ones that actually look like chloroplast when you're labeling. Same thing with this. This has not been chopped open. Neither has this one up here, these little bean shapes. So is that a chloroplast or is that a mitochondria? Like, what is it? We don't know. So don't label things that are vague and aren't clear. This is clearly a chloroplast. This is clearly a mitochondria. Label the obvious ones, please, so I can give you full marks. Okay. This one as well, they have not chopped open the nucleus because it was already shown in the other picture. But you need to know that inside you still have you have a nucleolus and you have the chromatin in there as well. And then also in this diagram, different than the animal one above, is you've got the Golgi body drawn very small. So just be able to label both of those ones. Now, we're going to get into cell transport after our lunch break. When we talk about cell transport, we're going to talk more about this word turgid. Some people say turgid with a hard G. Some people say it with a J, turgid. And it just means that this is full and pushing up against the cell membrane, which then pushes up against the cell wall. Also, when you're labeling this, make sure, I've tried to be very obvious with the red versus the blue here, this whole outside structure, again, because they've tried to show 3D, they've tried to show that this fat lip is the cutaway version of the cell wall, and then the thin line is the cell membrane. So if you could just be careful when you're labeling your diagram again as well when I'm testing you. So what's unique here? Well, again, water vacuole. You don't have this huge big storage site. It's not just water in there. There's all sorts of things being stored in there. The chloroplast, which is helping to capture sunlight in order to start chemical reactions that's going to give us sugar in the end. And then the cell wall as well, finally, is the unique thing here.